Hey, Jeff.
Hello.
Hi.
Hello, hello.
All right. Ready to talk about resource reports?
Yep. Let me update my. That's better. I'm not so small anymore. Okay, so let me know when I should.
Yeah, let's dive right in. You want to go to shared screen to
Yep, let me do that.
fantastic. And people can join as they come on. We had like 15 attendees, so we'll see who comes. Great. Do you want to do full screen on it?
Yep. Oh, sorry. Wrong thing. Wrong button. There we go.
So little time. Okay, perfect. All right. Well, welcome to our attendees for the third in a series of boot camps that we are putting on together with dkNET. And today we're talking about the dkNET resource reports, RRIDs in support of better science. And surprisingly, there's a lot to learn here. So I'm welcoming Dr. Jeff Greth from the University of San Diego and UC San Diego, sorry. And hopefully he's going to enlighten us. I'm sure he will.
Okay, thank you, Monica. So today I'm going to cover the resource resource identifiers and a number of the tools that we have built out around that in support of improving the rigor, reproducibility of research. But first, since I don't know if everyone's been in all the boot camps, just wanted to give a little overview of dkNET. So the NIDDK information network is funded by the National Institute of Diabetes and Digestive and Kidney Diseases at the National Institutes of Health. And we are really meant to be a information portal for biomedical researchers, both basic and clinical, and also serve as a way of connecting researchers in research related to NIDDK to resources that may be available to them, and also to provide support in linking some of the tools together. As Monica mentioned, we are the primary site for dkNET is at University of California, San Diego, and we also have collaborations with Baylor College of Medicine, as well as a number of other institutes. So dkNET has a number of different areas in the portal. There are a number that we won't be discussing today, such as fair data, data discovery, and the Hypothesis Center. Hypothesis Center will be the next boot camp in early September. So be sure to come back for that one. But today I'll be focusing on the research resource identifiers and the first two, the resource reports and authentication reports. For an overview of all of the dkNET tools, you can actually go to a prior boot camp, the boot camp on August 6th, where we discuss sort of the dkNET informatics tools and supporting the research lifecycle. And so again, when you come to the portal, there are a number of tools, again, that we provide. And again, on this left panel is where we're going to be focusing our discussions today. And so in terms of, what researchers are involved with in terms of, planning their experiments and executing their experiments, publishing the results, where the resource reports and authentication reports come in really is in this initial planning phase. And I'll show a demo where you can get more information about resources through the resource reports. And also in terms of generating necessary authentication information for NIH, for example, if you are submitting a grant application. And then at the end of your research life cycle, you're going to be publishing those results. And here again, we have the need to get into the resource reports to cite your RRIDs. and also potentially to track usage of resources. So again, we'll go over that a little bit more when I get to the demo. So we'll touch on those aspects. And if you're looking at specific questions, again, there are a number of tools in the other areas that will help you as well throughout this entire research lifecycle. So if you're interested in getting an overview of everything, please go visit the August 6th webinar as well. So how do you describe your reagents? So research, you may want to you can look at it sort of like cooking, in the sense that you have step one, you have the materials that you need, and then the methods that you need to use to actually run your experiment. Now in the literature, in what used to be a normal practice is that you would mention what you're using, for example here, a model organism, and then potentially where you got it from. But if you actually were to actually go to the Jackson laboratory and try and search for this, you wouldn't actually be able to find that. And so there's been an issue with the identification of reagents and resources used in the biomedical literature. And we've done a study in collaboration with Vasilevsky when we began sort of looking at developing the research resource identifiers, looking at how well are resources identified, based on at that time sort of the standard publication practices. And for antibodies, cell lines, constructs, I mean, you were basically lower than 50% being able to correctly identify what was actually used in a research publication, and that is critical if you're actually trying to either reproduce the work or use that antibody or cell line, for example, in further research that you're interested in. And so this is why we began working in developing research resource identifiers. And really these are meant to identify key biological resources. And so what are RRIDs? RRIDs are unique identifiers for these resources. They're search friendly, so you can actually take the RRID and go to Google, for example, Google Scholar, and search for anything that Google knows about RRIDs. And it's provided by independent community registries. So for each resource type, for example, antibody or cell line, or plasmids, there is a separate community authority that is developing the RRIDs for this. When we deal with model organisms, it really looks at the model organism databases that support that specific organism. So there are there's mouse genome informatics, which deals with mice. There's RDG, the rat database. So we're pulling information from multiple community authorities there in terms of the organisms. But basically, each of these community registries tracks the resources, and then the RRIDs are generated from those identifiers. And so when one looks at the anatomy of an RRID, and sort of how you can cite resources more explicitly, is you can still provide the company the local catalog ID for that, the version or lot used, and then the RRID itself. And so this is the sort of global persistent identifier for this resource. And why were RRIDs created? One was for tracking. So for individuals, labs, researchers developing resources, it makes it easy then to track these through the literature. to see where they're being used, but also for rigor and transparency, right? So really to explicitly identify what reagent, what resource was used when you publish your results. And these are now being included in a number of journals and are required and recommended. So, for example, here's Nature Research mentioning the resource identification initiative and examples of how these look in the literature. And there are a number of different requirements and recommendations. So, for example, cell presses star methods also include details on how to include RRIDs in their star methods. eLife, has been a big supporter as well. Society journals, so the Endocrine Society, for example, are all trying to improve the way that authors are citing the resources that they are using in their research. And so when you actually go to the actual companies that supply these as well, they also provide information about the RRIDs. A number of them do. So, for example, here, BioLegend in regards to antibodies, they provide the actual RRID as well on their catalog pages. Cellosource, which deals with cell lines and provides information on cell lines, also provides that information. So a number of the community authorities and also the vendors are starting to include these as well in their catalogs. In addition to sort of requirements and recommendations from the journals, there's also now, through NIH, for example, NIDDK and NIDA are, expected to register resources and also use RRIDs in their research. So this is also, coming through NIH, and it's actually interesting to look at how sort of the identification of antibodies sort of improved, based on when RRIDs were actually brought into the literature. And so, looking at sort of the time when we started, in developing research resource identifiers and began including them in journal publications, that was around 2014. And so if one looks at sort of the ability to identify certain antibodies, there's a large jump in terms of the identification over the initial 4-5 years of the pilot when we were beginning with the research resource identifiers. Now, anyone can actually register resources. So, for example, if you're a lab developing antibodies or you're developing model organisms, when you come to, for example, the RRID portal, you can actually get instructions on how to provide this type of information to the various community authorities. So, for example, for the antibody registry, you can actually go to the antibody registry, enter the information online, and you will get an RRID that is curated and will be available within 1 to 2 business days, for example. There are other ways that we generate, which RRIDs are generated. For example, if you're getting biosamples from a number of the biorepositories that are using RRIDs. So, for example, the Integrated Islet Distribution Program. These are actually registered through NCBI, through the NCBI Biosample Database, and so the IIDP does the initial registration, and then when you actually get your your samples, they have the associated RRIDs that can then be used in the publication. So the growth of RRIDs has been has proceeded very well. So it's they're in use in over now 1,500 journals. Again, major publishers supporting the use of RRIDs. A couple of years ago, it became part of the standard format that publishers use to transfer information to the National Library of Medicine and PubMed, and so that's the JAT standard. It's the latest checklist for authors and material design analysis reporting checklist are included in there as well. And they identify more than 70,000 distinct resources that are being used. Now, in terms of sort of discovering where RRIDs are used, we've also developed tools around that. So, for example, we have a tool called SciBot that actually goes out and looks at the open access literature, and we can actually pull out the RRIDs, and then these get curated so that we can actually pull out information about where RRIDs are mentioned in the literature. Now, with these identifiers, we're able to pull in a lot of information about resources, right? So, we have the information that comes from the community authorities. We can tie this, as I just mentioned, to the literature. We can also tie it to external data and information resources such as ENCODE, the Human Protein Atlas, International Stem Cell Consortium, and we can actually then pull in additional information. So, for example, if there's any validation information, if there's any information about problems associated with the resource, we can pull those in. We can also, based on the literature, we can pull in information about who else is using that specific resource, and then we can bundle that up and provide alerts, for example, to the researcher. And so this is just an example of some of the information that we're pulling from various resources around there. So, for example, for cell lines, we get information from through CellaSource from the International Cell Line Authentication Committee, that curates information about cell lines. And so, for example, misidentified or contaminated cell lines are identified within the system. There are other ratings and information, for example, provided by the Human Protein Atlas, again, the Intestinal Stem Cell Consortium, and others. And so the way that we bring this all together is through the resource reports, and the resource reports provide a way to view all this information. So on the report, you'll see that on the top there's the basic information about the resource. There's the specific information, including how to properly cite that resource and additional information. Then we get into the usage and citation information, as well as any ratings or alerts that we may have about that. And then also information about how to incorporate information into an authentication plan, and I'll talk about that after we are through with this section on the resource reports. And so when you actually click on this, you will actually see the usage citation metrics, and you get an information about the mentions. And so what I'm going to do now is going to bounce out of the presentation, go to the portal live, and let's see if the demos work and my internet doesn't go down. So if we click on the resource reports and click specifically on antibodies there, I can do a search, for any keyword that you would like to search for. There are two ways to actually view this information. One is in a table view, so it gives you, the ability to see the results in a table. You can actually, sort on these as well as downloading the results. So if you want to download, the search results you have, you can do that in table view. But what I'll do first is I'll do most of this through the snippet view, which gives you a Google-like view of the results that you've returned. So one thing to note is on the left, you'll have a set of facets that provide the ability for you to, for example, filter on host organism or, the target organism or the vendor. But what I wanted to really show, was the ability to see, okay, well, out of all the, antibodies that have been returned, which ones have been, which ones have we found more information in the literature about? And so you can see that when I do that, there are certain antibodies that come to the top. You'll notice also that some of them have this little green star. And so that green star basically highlights that there's either, some information about, some either validation information or information related to that resource. And then you'll also notice to the right of the name of the antibody, you'll see that there's information about authentication reports. So as you notice, this first antibody I already have in an authentication report. The second one I do not. So there are shortcuts here to also add your information to an authentication report. So for example, if I click on this, it will give me a list of my authentication reports that I can then either add or that I can add it to. And so if I actually will go and look at one of these, so I'll just choose the first one. Again, so the resource report starts out with, the antibody information. So if you expand that, you'll get, all the information about, the proper citation, information on, some of the references that cite it, the vendor information, and potentially other catalog numbers as well. If one looks at the full usage report, this will list the mentions that we have been able to extract out of the open access literature. And so the first part of the usage report is, the articles by year. And, you'll notice 2021 is not completed. So that's still, we're still gathering information on 2021. And also, since the open access literature is sometimes 6 months behind the primary literature in terms of what we're actually able to mine using the natural language techniques we use, this number will definitely increase over the next 6 months. So one of the things that we provide is, since we're pulling information about all the different types of resources used, you'll notice that there are additional antibodies, that have been used in the same publications as with this antibody. You'll notice that there are also image analysis tools being used. So for example, Fiji is an image analysis tool. And so you can also see, what other resources were mentioned. But then when you go down to the actual mentions, we'll list the papers. If you click on the PubMed identifier, so let's if I click on this first one, give it a second. So basically, This will show our view of this PubMed paper, and what it will do as well is, within this paper will tell you, for example, all the antibodies that are used in that paper, and also, for example, other research resources. So here, for example, they used ImageJ for image analysis, and so that gives you, specific information about the whole set of research resources identified by RRIDs in that paper. Close that window. So again, if I were then to look for city, so I'm at UC San Diego. So are any of these researchers local to me? And so I can actually filter the mentions and find out researchers that are in my area if I had, for example, any questions about the antibody, or if I wanted to contact those authors. And so this is another way to find sort of local expertise around research resources that you may be interested in. So let me go back to the resource reports in general. Again, and so right now we have a number of areas that we cover. So software tools, cell lines, antibodies, the model organisms, plasmids, the biosamples, and then also protocols. So protocols are publications of protocols through services such as protocols.io. And the main difference with the publication with these published protocols is that they're actually using digital object identifier. So the DOIs are also what are used for any of your journal publications. So when you have a publication that comes out in a journal, it has a unique identifier, which is the DOI. And so our resource reports support both RRIDs and the digital object identifiers for things such as protocols.io. And so if I get back into let's go back into, let's just go into cell lines. So I wanted to show you a little bit more sort of about some of the alerts, that we pick up. So again, on the left, we have the facets, that you can go through. And so, for example, I'm just, to make it easy to show you, there is information about the problematic cell lines, and this, we make this very explicit, when you come to the resource reports, so that if you're looking at information about a particular vendor and catalog number for a cell line that you're using, that that information is right up front. We can also look at what are the most commonly cited cell lines that have problems. So, for example, HepG2, which is well known, and that is misclassified cell line. And so here's the information from ICLAC about all that information. And so what I will do is I will also now go back to the antibodies. And with validation, there's a number of different places where we're getting validation information from. And so let me sort by using some of these facets to just to sort of give some information to show you what that looks like. Right. So again, this is down in the ratings and alerts section, and so this is where all those ratings and alerts will show up. And let me go back. One other thing I wanted to touch on is the community authority that we're pulling the data from is in this data and source information right here, which is the antibody registry. Okay, so I think I touched on a number of the features with with the resource reports, let me go back to my presentation. Whoops, wrong button again. Okay, there we go. Okay. And so, one of the things that NIH has, really been pushing over the last number of years is really, enhancing, rigor reproducibility, and NIH, has had released guidelines around this, and so there are, new mandates for rigor and reproducibility. One of them, relates to preparing authentication plans, as I mentioned before. So any new, grant submission needs to have authentication plans. If you are using, key biological chemical resources such as cell lines and antibodies. And then also, another part is managing and sharing data. So NIH has new guidelines coming in that take effect in 2023, where everyone, who is funded by NIH will have to, make their data available to the research community. There's actually also we have a boot camp from July that you can also look at that discusses sort of fair data and those new mandates. So in terms of the authentication plan, all grants, are required to include a one-page attachment, how the grantees plan to authenticate, the research resources, the reason for that is that, these resources, vary from lab to lab. They can vary from application to application. And you also, want to make sure, that the resource you're using is actually doing its job. And so, what is NIH trying to say with the guidelines? Again, pay more attention to the methods. Make sure that things are documented well, such as the RRIDs. But the other issue, and this is why also we're trying to pull all this information about the performance of resources such as cell lines, is that people are still publishing with contaminated cell lines, and cell lines that may be problematic. Now, in certain cases, depending on the study, the cell line used, the issues may not affect the research being done, but still, it is something that has to be looked at closely. And if someone is using a problematic cell line, really need to describe why it is not problematic for the research being done. As I mentioned, for cell lines, the International Cell Line Authentication Committee, really provides sort of the useful guidelines, how one should verify cell line, how one spot checks cell lines, and that when and where you should be doing this. So you should verify cell lines at the end of experiments, and you also should check with ICLAC with dkNET for information about the cell line, anything that may have come up since you started using that cell line. And so again, this is where the resource reports come in. This is something that I showed you in the demo, and you can actually click back through to the ICLAC Cellosaurus site, and get more information there. Now, one of the things that we looked at and was actually very interesting is that when we looked at publications that use cell lines and then compared papers where there was no RRID detected versus papers with RRIDs, we actually found that when authors cited RRIDs, that was associated with a lower reported use of problematic cell lines. Again, can't say that exactly why, but there are numerous reasons why that might be the case, in the sense of noticing that when you're assigning RRIDs that that a cell line does have issues, you may want to look at that research again. But the main reason for RRIDs and pulling this information is not to have this information available at the time of publication. When we really want this information is at the very beginning, when you're starting to do your research. And we have the same issues with antibodies, where we have issues with cross-reactivity, variability, and also using antibodies in techniques that they're not supported in. Also within for antibodies, there are actual guidelines in terms of what are considered validation pillars for the antibodies, in terms of how one should authenticate the antibodies themselves. And there are a number of types of information that you can pull from from the antibodies. So for example, antibodies in the resource reports, so even if the same antibody is sold by 3 different companies, there's one RRID to track, that same antibody across those companies. But you also have, information, about, that antibodies were discontinued, what the manufacturer provided, applications it was tested against. So, there's additional information that we're pulling in, around antibodies. And, as I just mentioned, we really want, people to, researchers to, look at this information, as they are getting started. And so this is why we created a tool around the resource reports and the RRIDs, and these are the authentication reports. And really, we want to alert researchers of any issues, before a study is performed. Also to provide guidance on, the authentication methods. So, for example, here is, just an example of an authentication report. And so the report itself that gets generated has information on the cell lines and antibodies. As part of this, there's a little table that has information about the antibody, which has its RRID, the vendor catalog number, also has comments, and then also any alerts that we may have. And there's also then, the information in that section about, potential validation. So what I'll do now is I'll actually go into the authentication reports. So let me get to my other window. Okay, there we go. So here, instead of going into the resource report section, we'll go into the authentication reports. And even before you do anything in terms of adding antibodies or cell lines, you can get this basic information on cell line authentication. So here, there's a little guide in terms of how to check the cell line information that we get from ICLAC through the resource reports, and then goes into some other information, and then, provides the links, for example, to NIST that has information on standards for cell line authentication, provides the cell line checklist from ICLAC. And we do the same thing, with antibodies. going in, checking, your antibodies through the resource reports first, but then we also provide, the validation pillars, and, provide references, to that as well down here. So, giving you information on how to do that. But if you, get started, click on the start here. So the resource report, the authentication reports have a dashboard, and so you'll have, a number of your reports here. And so if I click on a specific authentication report, it will basically have on the top right, we'll have the listing of all of the items that you have added. You can actually do a search in the authentication reports as well. So if I were, just to search an antibody, for GAD65, I could search that down here as well. And then I could either click add, for example, if I wanted to, and then I could also remove them from the report, hitting that same button. So again, you can select the antibody cell line, search what you want to do. If you have a vendor or a catalog number, what sometimes is a lot quicker and more direct is if you actually, so let's see if I clear this, get rid of that. And let's say I'm doing bio and do let's see if I wrote down that correctly. Yep. And so that will be, really the one item you find. So using the vendor and catalog number search is, really reduces the number of results and should target, the specific resource that you're using. And this is, also available in the resource reports as well for you to do a more focused search using that. So again, you've collected a number of resource report items, in your authentication report. So if you can then go to preview of the report. And again, you'll get a listing broken down. So first the cell lines. And so, for example, also, sex as a biological variable, which is something NIH is focusing on. So we also, provide information on sex as well, and then information on the antibodies, and this information, and then the resource, the full information from basically the resource reports, the main information provided at the end there. So once you have a report, you can actually save the PDF of this report. So this will create a PDF. It will actually, store that, and then you can come back later to look at that. And so, I can now, download the PDF, download the PDF itself. One important thing to note is that you'll get a list of PDF reports that have been snapshotted. The reason why we allow the different time points for snapshotting is this is actually a live dashboard. And so when you're actually doing your experiment, you can actually come back 6 months, down the road, and you can actually basically go in and check again, if any alerts have popped up. You'll see the little red triangle or a little orange symbol, and you can then again snapshot the report at that time, if that's something that you'd want to do. So again, I've sort of covered the resource reports, the authentication reports. Again, there's a lot of information that's available through these, but we're sort of running out of time. So what I wanted to do is just a couple more slides I want to get through, and then we'll be able to take some questions. So really, what we're really trying to promote is the use of RRIDs in publications, also in grant applications, wherever they're necessary to uniquely identify the resources that are being used. If you do review papers and grant applications, double check the methods, make sure people are being very specific about the resources they're using. So we're really trying to make sure that the scientific literature becomes much more transparent in terms of the resources that people are using. One thing I did want to touch on, so we work closely with a number of different projects and groups, and one of them actually is protocols.io. And so, as part of the D challenge, with sugar science, you'll notice that, we're working with protocols.io, and I mentioned it earlier in the presentation as well. So this is a site where you can actually publish. So it's an online platform for developing and sharing reproducible methods. One thing to note, so there are a number of researchers, research labs, already publishing protocols here. And the nice thing about publication of protocols here is that, as we mentioned earlier, there are two primary components, to be able to, do a scientific experiment. One is identification of the ingredients. The other is, the documentation of the methods. And in journal articles nowadays, there isn't, that much room to fully describe everything necessary about a method. And so protocols.io allows a full, complete, step-by-step description of a protocol. It also allows association of the research resource identifiers, with your reagents and resources used in the protocols. And so, this is a very good way to really provide, very transparent methods. And again, they do get a DOI, as I mentioned earlier, which means that your protocols can also be cited directly in any other publications being made. The other thing to note is that there are also a number of large consortia, from funded by NIDDK, the Mouse Metabolic Phenotyping Centers, DICOMP, HERN, the Integrated Islet Distribution Program, also large NIH Common Fund projects such as SPARC, are beginning to really publish a lot of protocols, that make protocols.io a good repository, to look for protocols that might relate to your research. And so that is basically what I wanted to cover today, and I think I have left enough time for questions.
Yeah.
Any comments?
Hey, Jeff, that was great. We do have a couple questions in the chat, and here's one of them. I can't find the antibody that we used in my lab in dkNET. What should I do? Should I contact the antibody author?
So the most direct route that you could take is actually, as I mentioned earlier in the presentation, you can actually go to that actual community authority. And so in that case, it is the antibody registry. And you can actually request the registration of an antibody. So you can actually supply the information about the vendor, the catalog number, and then the curators at the antibody registry will actually look to see, if that actually does exist, and you just and wasn't able to be found, or if it is something that isn't in the antibody registry, it will get added, and you will get an RRID that you can use.
Okay, that's a good answer. What if
so I just want to say, and it's also the same for the other community authorities. So, for example, for model organisms, you would contact the specific model organism authority. For example, contact RGD or others, to register a model organism that even you may have developed, right? So that you can register with that authority.
Yeah, that's a good qualifier. What if, another question was, what if any publications are requiring RRIDs for publication currently?
A number of so I mentioned sort of anyone that's using the MDAR checklist and the STAR methods, for example, those are sort of new guidelines for how to be transparent in providing that information. And those are being required, by eLife, CellPress. I believe the MDAR methods are going to be used by Nature and Science. So, I mean, a lot of these sort of new reporting checklists, include, the need for RRIDs and are being, picked up by, many of the publishers. And so,
So it's definitely time to get on board with these.
Yeah, and then it's
tools.
also, I mean, again, it's also better for science, right?
Yeah, absolutely. And then, is there any NIH incentive around the use of these RRIDs? Deadlines coming up. I mean, what is the take on that?
Well, I mean, I think the major sort of incentives requirements are coming from the publishers. As I mentioned, there are, within NIH, there are some institutes now that are starting to require that, you register your resources. I mean, the same thing with material sharing, for example, for model organisms. If you register your model organism with, the community authorities there, you're meeting your sharing mandate requirements, plus also, they'll have their RRIDs associated with them.
Yeah, so it really is a matter of just getting comfortable. dkNET is a really great portal to explore and get comfortable with these resources and get yourself armed and ready basically for all the changes that are happening, good changes in the publication world and in the grant writing world. And I think that this really demonstrates that it's just a better way of doing business for scientists. And it's one that everyone has to get more comfortable with. It's not, it's still somewhat new. And so it's so important that, you guys sort of take the time to share, why it's important and how to get access. And we really appreciate you coming by today and doing that.
Oh, no problem. And I think, the main thing, that we're really trying to do is help researchers, basically enable them, to do better research in ways, in the sense that, you're able to, more easily discover, information about the research resources that they're using, be able to better decide, which is the most appropriate, antibody cell line, and also be able to track, in the literature, the open access literature that we can pull this information from, and also from the community authorities, if there are, any issues, or, looking at, that specific antibody, in the literature, as to what others have, what other researchers have used it for, or any other, information that might come from, the use of that specific antibody, something that was very difficult to do in the past, because you weren't able to easily search, for that antibody in the literature. But with the identifiers, you're able to actually do that in a very straightforward manner, and allows you to collect, a lot more information about the resources that you're using.
Yeah, and as we know, antibodies are, can be particularly finicky and hard to, some of the data around them can be hard to replicate. So this is a very important piece. And I guess we'd just like to say, from the Sugar Science, we completely champion your dkNET's mission of facilitating, accelerating, and really authenticating data and helping scientists do their job. So thank you again for meeting with us and talking with us.
Oh, you're welcome.
All righty. Signing off till the next one. Look forward to it. And that next one's coming up September. And I believe that one is going to be all on September 10th at 11 a.m., all about the Hypothesis Center. So we're waiting for that with sort of bated breath. It's going to be exciting. We'll talk to you later.